So I am honored to be here today with Dr. Adejoke Obajuluwa, who is a scientific researcher and a university lecturer at Afe Babalola University, Ado Ekiti, in Ekiti State, Nigeria. She studied cell biology and genetics and has a master's and a PhD in the discipline from University of Lagos and University of Ilorin. She has a number of scholarly articles which border on genetics, neuroscience, and cell biology in journals both locally and internationally. And she specializes in molecular toxicology and is passionate about education for informed health choices. And the research that we're going to discuss today is her and her team's research on radio frequency electromagnetic waves on biological systems. And the research that she has done and with her colleagues is on Wi-Fi radiation as well as other exposures. So thank you so much for being here today to share your research findings with our community in the United States.
You're welcome. I'm excited to be here.
So tell us about your department and the research that you have performed and your findings.
All right, thank you for this opportunity to share our research findings. And not just that we have this platform to do this, but so much more that at this time, people need to know what the effect of radio frequency electromagnetic waves exposure have on biological systems. And I would like to first introduce and talk about my research team and how I came about building this team for this research. Afe Babalola University is located in Adejoke, Ekiti State, Nigeria, and it's a private tertiary institution. That's where I work. As a lecturer, I teach genetics and specialize in molecular toxicology, cell biology, and neuroscience. So we are tasked with, to investigate and go into research areas that have not been so well researched upon. Like we have a call to do what a lot of other universities are not looking into, because the university's aim and vision is predicated on the need to provide useful scientific results to the society and economy and a society and the nation at large, so we can make useful impact. So I sat back one day to think of what the effect of radio frequency electromagnetic waves would be, because we are increasingly, exposed. We have an increasing exposure to these waves based on the demand for internet in our place of work and even at homes. Here in Afe Babalola University, we have a data center where we have installed Wi-Fi devices and routers that serves the university community. So since we have such resource, I thought of talking to one or two people that will work together. I work in biological sciences department, basically. That's where I'm domiciled, and in the biotechnology unit. So I called a few friends of mine. I'll start with Mr. Olakunle Afolabi. He's specialized in toxicology and nanomedicine in the biochemistry unit of the chemical sciences department of the university. And also Dr. Ayodele Akiyemi, who is specialized in immunology and biochemistry. And Mr. Tibalola Madok Obajuluwa, who is a specialist in new media and health communication. And the head of the lab, who sits as a professor and the head of Department of Biological Sciences in Afe Babalola University, who is Professor Pius Abimbola Okiki. So we made up the team of researchers for this research. And the first thing we did was to sit back in 2015. We looked at review articles and we researched to see what the news has been, what the findings has been about the effect of this waste on people and in different biological systems. Then we went ahead to design our own research, to design a model to study this effect. So we would be sure that what we are exposed to on a daily basis is not going to affect us on the long run. So I'm just going to start the presentation so I can go into the findings of the research. Can I go ahead?
Yes, please.
All right, thank you. So now, like I said earlier, the curiosity was more out of the increased use of Wi-Fi connected devices for execution of daily activities across different cultures and populations. And this radiation emits energy in form of waves or speed particles. Now, it is said in literature that this energy is non-ionizing. It means it does not have ionization effect or does not have ionization potential, meaning that it would not have so much deleterious or harmful effect on living systems. But the research and the statement and press release by WHO in conjunction with International Agency for Research on Cancer 2011 actually raises doubt about this claim because they classified radio frequency waves as carcinogenic, and this brings some sort of concern, and we thought to investigate and find out if this is true. So the aim of the research was to investigate biological and neurobehavioral effects of 2.5 to 3 gigahertz band of radio frequency electromagnetic waves, and we used a rat model we used rat as a model of human health. So we have the objectives here. We evaluated the cerebral cortex acetylcholinesterase activity. Now, acetylcholinesterase is an important oxidative stress biomarker, and it's also an important biomarker in environmental health. It has been the focus of so many research. So that was why we picked that biomarker. We decided to look into this biomarker. So we investigated the mRNA expression levels of ACHE in the RFEM exposed rats. We also investigated the locomotor function. Now, this is how the rats are able to move. I will explain more on that so we understand that better as I go on. Then also we investigated anxiety-linked behavior in the rats that were exposed. Then our second objective, the second phase of the study, we went ahead to assess the effect of 2.5 to 3 gigahertz band radio frequency electromagnetic waves on the rat's brain through DNA fragmentation, profiling, DNA fingerprinting. The marker assisted selection we used there was the rapid amplified polymorphic DNA, and the random amplified polymorphic DNA, and we used the rapid technique and also histomorphometric analysis. We also examined the lipid peroxidation, hematological parameters, and key serum and tissue enzyme alterations in those rats. Now, this is the method showing our setup and the design. First, we had 24 male rats divided into 4 groups of 6 in each group. We put them in plexiglass cages 50 cm away from the Wi-Fi receptor antenna in the data center in the university here. And we varied the exposure. The group 1 was not placed in the same place with the exposed rat. We had a glass bar separating them from the ones that were exposed. So we had the control group, which had no RFEM exposure. And the group 2 to 4 was varied from 4 weeks to 8 weeks exposure of RFEM waves. So For the experimental procedure for the line crossing for locomotor frequency in the rats, we used a standard method by Bowen et al. 2014, and the method is such that you have an open field box that is 4 by 4 by 40 cm, and the floors are divided into 16 equal size squares. Now, the frequency, that is the number of line crossing motion or movement the rats make in the box, is videotyped and saved. And also we measured their anxiety in the box. Now, anxiety is measured through the number of times they defecated while in the box. We also measured their anxiety levels through the times. Let me show the video so we can understand. I'll go back to the slide later. I want to show the video.
Excellent. I'm glad you have the video.
Let me quickly do that.
And this is a Wi-Fi frequency and exposure.
Yes, it is.
Yeah.
I'm sorry, I'm trying to look for my stop share. Okay, yes, perfect. I have it here now. I hope you can see my screen. Let me take it back to this beginning.
Yeah, that's fine. I'll edit this, so we'll just start at the video.
Okay, can you see the video now?
Yes.
Okay, perfect. Now, this is one of the rats. This is the open field test box, like I explained earlier. And the rat is being monitored while it's moving in the box. We can see the squares, and we can see how the rat is moving around the box. These black spots are feces from the rats, and they are indicators of anxiety. Increased defecation is indicative of anxiety in the rat. So we counted the number of times the animal defecated while in the box, but more importantly, we counted the number of times the rats migrated, crossed these lines from this center point. And also, I'm trying to take it forward to the time that, yes, to the time that the rat is standing on its hind leg. Now, this is called hinding. What this animal is trying to do here is an escape instinct in animals. It is expected that an animal or a rat that has good cognitive function would make escape attempts by standing on its hind legs in order to escape from the box. So I'll just leave this place so I can go back to my slides. Okay, so can you see my slide now?
Yes.
Okay.
You can put it on presentation.
Okay, my presentation. Okay, so I just took out that time to explain what we did to assay for the line crossing in the rats, and also we placed them in the rotating rod. Now, this is typical for evaluating motor coordination, because this instrument, this apparatus that you see here, this equipment, is set at 34 revolutions per minute. That is, it's a rotating platform, and we place the rat on either of the spaces between the rotating rod. It is normal that the animal would try to hold on in an attempt not to fall off from the rotating rod. So an animal that displays a good holding on attempt is said to have, reduced anxiety. And an animal that falls off within a limit, within a short period of time, actually shows anxiety related, is indicative of anxiety. So we did that, and also we did RNA extraction, cDNA synthesis, and all of that. I don't want to spend too much time talking about the methods, so we can see them later. I'll just go straight to the result. Now, what we found out after 8 weeks of RF EMF exposure was that the number of line crossing, or the number of lines crossed in the control rats, was significantly higher. As we can see, when we compare this in the control. The group 1 is the control group, and the one that had the 8 weeks exposure, the group 3 had 6 weeks exposure, and the group 2 had 4 weeks exposure. We can see distinct progressive decline in the number of lines crossed in this rat. Also, I didn't put in the data for the defecation and hiding attempt because of the want of space, but we also noticed increased defecation in these other groups, in the RFEM waves exposed groups. So on the rotarod here, we observed as well that the group 1, which is a control group, stayed longer on the rotating rod than other groups, and it was quite distinct from how long the other groups, other rats were able to stay on the rotating rod. And this is indicative of motor coordination and anxiety in the RFEM waves exposed rats. So for the acetylcholinesterase mRNA levels, it was standardized against beta-actin standard for assaying for the expression, and we found hypermodulation. We found increased levels of mRNA ACHE level expression in the RFEM waves exposed rats. And we can see that it was progressively increased in the exposed groups when compared to the control. The same, it was vice versa in the activity. However, we noticed a good increased ACHE activity in the group 1 and a progressive decline in the acetylcholinesterase activity in other exposed groups in a similar pattern. And the control group was distinctly different, was distinct from other exposed groups. So the control rat exhibited high exploratory activity as seen here, and also they exhibited higher passive rotation and latency of fall from the exposed rats. We also noticed progressive alteration of acetylcholinesterase mRNA expression levels and activity in the RFEMW waves exposed rats. Now, what are the inferences we drew from this? Based on our findings, we can infer that neurobehavioral indices that we measured indicate that the RFEM waves induced stress in these animals. And the stress-related behavior, like I said earlier, defecation, increased defecation, reduced hiding, and also inflammatory responses with increased exposure plan. Now, the implication of this is anxiety and stress-related health problems. When we extrapolate these findings for human studies, is that increased exposure can cause or modulate anxiety and stress-related health problems with prolonged and frequent RFEM waves exposure. For the acetylcholinesterase activity, like I said, the acetylcholinesterase is a very important and a key biomarker for oxidative stress and a key biomarker in environmental health. So we noticed the activity was inhibited in the RFEM waves. And what this implies is that when there's an inhibition of this neurotransmitter activity, there will be an overactivation of acetylcholine in the central nervous system, thereby resulting in delayed response. As we can see from this diagram, we can see the receptor, we can see reduced enzyme concentration here, and it will result into poor memory and other cognition deficits. So I would like to cite the work done by Campanari et al. 2016, where they stated categorically that Alzheimer's disease is characterized by a decrease in the enzymatic activity of the enzyme ACHE. So it means that the exposure to the radio frequency electromagnetic waves is actually neurotoxic and can cause such, disease or disorder in biological systems. Now we have we also saw that there is high level of the mRNA ACH in the exposed RFEM waves rats. Now, studies have shown that there are high level or recorded high level of mRNA ACH in in tumors from human samples. Now, this is human population. They worked on humans, and they found that there was high level of ACH in astrocytic tumors. And this suggests a link between radiofrequency exposures and various brain tumorigenesis. That's not surprising because ACH is indicated in cell proliferation, and we understand that anything that tampers, anything that affects or dysregulates a cell proliferation system would have a downstream effect, a cascade kind of effect on other cell metabolic processes. So I'll go forward from here to the second stage of the work, where we also assayed for, we looked at DNA damage from cerebral cortex, and we analyzed this. We separated the DNA extracted from the cerebral cortex in a 1.5 weight per volume agarose gel, and it was viewed with a gel imager. Also, we used the rapid technique for DNA fingerprinting, the random amplified polymorphic DNA, to look for mutation patterns, to investigate mutation patterns and possible aberrations that could have been steered or spiked by the exposure to RFEM waves. Then we also did histomorphometric analysis of the rat brain sessions with focus on the cerebral cortex of the rat. And we made the findings. We found degradation of the genomic DNA. You can see the smear. The smear here is indicative of necrosis, and this an apoptosis. This smear is indicative of an acute toxic response to the exposure. And we found that when we looked through the profiles from the 4 weeks exposed rat to the 6 weeks and the 8 weeks, we would notice the thinning of the bands as exposure progressed. Even before the DNA fingerprinting, we could see from the genomic DNA profiling that some form of damage has been done to the DNA. Then we went ahead to look at the profiles. We used 6 rapid primers. They had 10 map primers, 10 oligonucleotides for this study, and we discovered, I'm sorry, the lane is not so clear, but this lane is for the control. This is for the 4 weeks exposed RFEM rats. This is for the 6 weeks, and this is for the 4th week. Now, this first 2 rapid primers showed more distinct bands than the other 4. So I'm just going to focus on this, although some inferences can be drawn from the last 2 as well. We can see that for the control group, you can see we can see that there was minimal fragmentation. You can see much more the fragmented bands in the other exposed rats. And when we look at other profiles too, we can see clearly that the bands were much more fragmented than the control group, indicative of apoptosis and DNA damage. Also, in the histomorphometric analysis, this is the control group. We can see normal architecture composed of nerve cells, the arrow and glial cells. In the group 2, we observed a mild vascular congestion. We can see the arrows there showing mild vascular congestions here. And the brain, there's supposed to be the brain is not to have the blood-brain barrier mechanism. by which blood doesn't flow into this, the vessels are not congested with blood vessels, and we can see engorgement of these nerve cells with blood. We also went ahead to investigate the group 3, that is those that were exposed for 6 weeks. We observed that the nerve cells had mild congestion and neuro via arrow, but some of them appeared as in control, but there were mild congestions in our observations. Then the third group showed severe vascular congestion, as we can see here. So we can deduce from these findings that there was damage to the brain DNA due to the exposure to the electromagnetic frequency waves. And from time immemorial, DNA damage has always been a biomarker for brain damage. DNA damage we've always used DNA damage as a biomarker for brain damage, and we could see that established by a study, Schwab et al. 2019. Now, we also noticed that the RFEM waves induced mild to severe with increasing exposure, mild to severe DNA damage, brain DNA damage. So with much more, with increased exposure, there was more damage with increased exposure to radio frequency electromagnetic waves. And this confirms its neurotoxicity, and it's linked to other brain-related health problems. Now, looking at that, we may be wondering why this is essential. Now, the DNA stability is essential to maintain cellular integrity of all living organisms, because every cell has its own genetic blueprint by which it operates. So if any alteration happens at the nuclear level, that is to the DNA in the nucleus, there's going to be a ripple effect of that in anabolic and catabolic processes. And as we know that the gene expression is regulated by, there's a central dogma for a gene to be expressed, and that is DNA to RNA to protein. So if anything goes wrong at the DNA level, then we are right to say that there will be some sort of problem at the transcription level, which is the RNA level, and at the translation level, where this transcript, the mRNA transcript, will be translated to proteins. And this suggests that there could be a lot of problems that would arise from increased exposure to radio frequency electromagnetic waves. And that's why we have this. This is from Karolinska Institute. I just put in this schematic diagram so that we understand now some of the effects that we saw in the gel electrophoresis profile that we saw here, and here can be explained using this. We can see single strand breakage could occur as a result of oxidative stress induced by the radio frequency electromagnetic waves, and other effects could be base mismatches. Now, when there's a base mismatch, we could have variety of chromosomal aberrations and disorder, genetic disorder. And also, we can see cognitive impairment, which was confirmed in our behavioral studies, and necrosis, then cell death. This suggests that exposure to increased levels would actually have a neurotoxic impact on biological systems. I mean, exposure to increased levels of radio frequency electromagnetic waves. We went ahead, the third phase of this study, to investigate a critical biomarker that is known for investigating oxidative stress. Now, when we say biomarkers, they're biological, we can measure this in biological fields, in biological tissues, like we did using the DNA from the brain of the rat. There are biological markers we look out for. The NDA is a crucial oxidative stress marker. So that was why we investigated this in the exposed rat. We also looked at the total protein, the AST levels, the ALT levels in the serum, and harvested liver, testes, and heart of these rats, so we can draw useful scientific conclusions. So we use standard laboratory protocols, like stated here, and the white blood cell, mean cell volume, mean capsular hemoglobin, and so on. But I just want to go on to the results right now. So this table right here shows the hematological parameters we found in the control and the electromagnetic frequency waves exposed rats. We found an increase in the mean cell volume of the exposed rats, and we discovered also that there was an increased mean cell hemoglobin, in a progressive increase actually in the exposed rats when compared with the control. If we look through as well as with my cursor, I have my cursor placed here, we can see abnormally increased packed cell volume. I must state here that the rats that we used here were 4 weeks old. So the standard of the PCV level for those rats, based on standard PCV levels that is published and standardized for Worcester rats, it ranges between 28 to 33 percent thereabout. But we can see that with increasing exposure, there's modulation in the level of the packed cell volume, and it's known in studies that abnormally increased packed cell volume is indicative of increased cell proliferation. And when there is loss of cell proliferation, there is a tendency of carcinogenesis, tumorigenesis, and other health related problems. So I would leave the hematological parameters now and go forward to show us the MDA levels. We can see the control here at a minimal level, about 4.5 micromole. But when we look at the exposed rats group, we can see increase, especially between the acute, the 4 weeks, we get into the 4 weeks response as an acute, and the chronic level. We can see the hike in the MDA level, and still hiked anyway, but just a bit drop from what it was at 6 weeks. And we can see the same pattern in the testes and in the heart of the rat. Also, in the protein, we found increased levels. This pattern actually conforms to the same pattern of the MDA, although this pattern in the testes was a little bit different from what we had here. It was relatively stable, but at some point at 6 weeks, it was modulated. And here we can see how the protein levels dropped in the exposed groups. Our conclusion from this study is this, that the radio frequency electromagnetic waves induce reactive oxygen species, leading to increased acetylesterases, mRNA levels, and activity inhibition. Now, the effect was not just that. We also saw the deleterious effect on the brain. We saw the deleterious effect on the genomic brain of the rats, and genomic DNA from brain of the rats. We saw DNA damage. We saw bands that were not initially present reappearing here in some of the other rapid primers, which is indicative of chromosomal rearrangement and can also be very dangerous and harmful to health. And we also found lipid peroxidation in harvested tissues, the liver, the heart, and the testes of these male rats. Now, we would wonder how this 8 weeks exposure was able to induce such great effects. And so I found this from Biochemical Journal 2011. It actually is a schematic representation and actually summarizes our findings. So it informs my choice for putting this here. Now, when we talk about oxidative stress, the molecular targets are usually the membranes, and we can see lipid peroxidation as found in our study as well. We could observe that reactive oxygen species had significant effect on the membranes, which led to lipid peroxidation and damaged membranes and lipoproteins. Leaving that aside, when we go forward to the effect on the DNA, we could see that there was DNA damage, DNA breakage, and mutations leading to cancer. Also, we found that the mRNA ACHE levels were significantly altered. They were hypermodulated, and this has been indicated in astrocytic tumors. There has been indication that there is hypermodulation of acetylcholinesterase, which is a crucial neurotransmitter in living systems, in all biological systems. And this suggests that there could be progression, or there could be disease, cancer and different pathological problems, coming up or arising as a result of exposure to radio frequency electromagnetic waves. So it's important that regulatory bodies at all levels ensure adequate research to ascertain safety and strict monitoring of safe limits and levels before deployment of radio frequency electromagnetic waves emitting technologies. I was able to listen to the vice chairman of the Nigerian Communications Commission, Professor Professor Dan Bata, where he stated that they had a 3-month trial of the 5G network deployment in some locations in Nigeria, but they altered the deployment of 5G technology after the trial, the findings from the trial. So we've been able to establish through this research the neurotoxic ability, the modulating effect, the deleterious effect of exposure, especially increasing exposure to tech devices that emit electromagnetic waves. So some of the tips able to put together is we should avoid holding the cell phone close to the body. We should avoid carrying mobile phone in our pocket and the belt close to the body whenever it is powered on. Now, it is important to note that our laptops are not to be placed on our laps, even though it is called laptops. It shouldn't be placed on our laps. It should be placed on tables or hard surfaces that are, a bit distant from our body to avoid the absorption of these waves into the cell membranes in our body. Then it is important to turn off our mobile devices when not in use or switch to airplane mode. Now, based on our study, we decided to recommend for further research, which we are interested in as well, and we hope to embark upon with the help of research grants, both here in Afibala University and internationally. There's need for public engagement and education on the safe use of tech devices, because virtually everyone on the road has 1 or 2 or more tech devices strapped on their bodies, ranging from wireless wristwatches, wristbands, to wireless headsets, to different tech devices that are Wi-Fi enabled, and it calls for the need. There's a need to engage the public and educate the public on how to use these tech devices to avoid health problems. Also, we're hoping to look at association study of brain tumor genes and proteins modulations in RFEM waves exposure. Since we have a functional data center here in the university, we are trying to see and investigate other brain tumor genes so we can draw conclusions, even though we were able to conclude, but so we can actually have a more robust conclusion on the effect that radio frequency and electromagnetic waves, predispose, or the harmful health effects or health conditions we can be predisposed to through constant and increasing exposure to them. Then we are trying to look at epigenetic memory remodeling capacity. Now, we have been able to establish the genetic, let me say the genotoxic capacity, genotoxic remodeling capacity of EM waves. However, we understand that anything that happens at the nuclear level occurs both in the somatic and the germ cells. So there's possibility that even the somatic cells, there could have been some form of aberrations that can be faithfully replicated in subsequent generations. So this is like we're trying to look at a transgenerational effect of EM waves, and we hope to graduate from using a rat model to taking it to places that have high density or places of high exposure to electromagnetic waves to be able to draw our conclusions. In conclusion, I'd like to acknowledge the founder and the pro-chancellor of the university who granted the funds for this research, the vice chancellor, Professor Smaranda, and Professor Adejoke, who sits as the provost of the College of Sciences, and Professor Amos Onusoya, who is the head of the Department for Chemical Science. And much more important, I'd like to thank Dr. Yinka Ajiboye. He's a radiation physicist, he's a staff, he's a lecturer here as well in Afe Babalola University. And I'd like to thank the ICT department and the head of the ICT department, who is Dr. Bayo Gundipe, and also to acknowledge the TWAS research grant we got for publishing one of the papers in toxicology report. Thank you.
This was absolutely an excellent presentation of this critically important work, and I'm really honored that you could provide us with all of these details. So did you do measurements of the radio frequency levels in the room?
Yes, we did. We used the EMR 300 probe to measure the wavelengths in the room, and we did that with the help of Dr. Yinka Ajiboye that I mentioned when I was acknowledging the people that also contributed in this research.
Many health ministries in Europe have recommendations to reduce exposure that are part of the Ministry of Health's recommendations. They say, keep the phone away from your head, protect children. They have all of these recommendations. Does Nigeria have any of those?
To my knowledge, there is none. And this informed why there was a video recording based on our research findings, especially when there were a lot of agitations. We found out that the public is not even aware. Very few people know about the effect of these things. There is no public awareness program. There are no public awareness programs. There are no agencies, making mention of this effect and trying to educate the populace on safe use of tech devices. So we came up with a little YouTube video.
Oh,
Yes, yes, and in our little way, we just try to share and enlighten people in our own sphere. But that's limited to the number when you go about several millions of people. When you compare the number that has seen the video, I think we had about 200 or 300 views, on the video, but it's nothing compared to the population of the Nigerian populace. It's quite large when you compare, when you take out 300 out of millions. It's just like a drop of water in an ocean. And we had to do this because we could see a lot of ignorance and lack of knowledge, and we were triggered to do this because we were also confident to do that because we did the research and there were profound findings. So I think we were able to put that out confidently based on our findings, on our research findings. And aside that, I also, like I told or mentioned earlier, that before we embarked on this project, we found that I found that there were limited research findings and research papers from Nigeria. A lot of countries around the world have done a lot of work in that regard. I mean, talking about radio frequency electromagnetic waves effect. But we had very few, in fact, would I say few? At the time of the study, there was none existent at that time. There was no research activity or research paper stating such activity. So I'm not surprised at the limited knowledge, or when I say the inadequate public engagement and populace awareness, because of the paucity of data from Nigeria. So I'm not surprised at that. So I think that's just my talking on that.
What about 5G? You may be wondering how the fifth generation wireless network would affect human beings. This is because people all around the globe are largely dependent on the use of tech devices for execution of day-to-day activities. This includes the devices we use at home, our handheld mobile phones, at school, in our places of work, for personal and social purposes. The list is endless. Now, the 5G is intended to overcome the enormous growing demand for data and internet connectivity that comes with this present IT age for seamless communication and speed. But we must consider the health consequences of taking such actions. So let's take a look at some of the research outcomes of a project study where we used the rat model to assess the effect of wireless network radio waves on living systems. Radiation from the wireless network is classified as the non-ionizing type, and so it is considered or perceived as harmless. But scientific evidence raises doubt about this claim. WHO has also classified it as an environmental pollutant, coupled with diverse scientific evidences from around the world, and the upcoming slides shows some of this reasons for this concerns. Assays that were used to ascertain the safety of the existing 3G network in a span of 8 weeks. The behavioral tests were done to assess for damage to motor coordination, to assess anxiety levels and exploratory ability in the rats. The brain DNA damage was also examined and acetylcholinesterase gene was assessed. This gene is critical for memory and learning. Hematological, biochemical, and histopathological profiles were also assessed. After the experiment, which lasted for 8 weeks, exposed rats showed brain DNA damage, acetylcholinesterase gene impairment, altered hematological, biochemical, and histopathological parameters. All this were induced just after 8 weeks exposure. The findings of this research was presented at the Women in Science Without Borders conference in 2018, and some recommendations were made. It's shown on the slide right now, but these findings suggest that exposure to higher frequency signals such as 5G would predispose living systems to a range of minor to serious health issues and therefore cause for concern. The links to the full papers are on the screen.
Do you have funds to continue this work, to do the follow-up recommendations which you're talking about?
We require funds actually, because even though the university make available some fund for research activities, the level at which we want to investigate and really take this study further, the university may not have that much capacity for it. So we would appreciate collaborations and funding from agencies that are focused on this research, and we would really love that.
It's always a challenge to get adequate funding for this.
Exactly.
Really some of what we would consider to be the most important research, because it's not adequately funded in any country that I know of. There's a study that was published relatively recently that found thyroid cancer linked to certain genetic susceptibilities in people, and that was done at Yale.
Yes, we are aware that there's certain genetic susceptibility, especially in schizophrenia, that has been established in studies in Alzheimer's disease. We understand. So we are trying to investigate candidate genes and that are indicated in cancer, brain cancer, so we can draw a much more robust conclusions based on our studies. We actually placed the rat at the center. The movement of the rat is monitored from the center where the cursor is placed on right now, and we can see that the rat's movement is also monitored in the box to see activities like crossing of these lines. We count the number of lines this rat has crossed, and we also check the number of times this rat defecates while in the box. It is important to state that this was done monitored for 10 minutes. Normally, it's usually done for 5 minutes, but we thought, given much more time for the rats to acclimatize to the box, we'll rule out every form of bias that may come from using a short period of time, a short observation period. So we observed this for 10 minutes, and we also looked at how many times the rat stood on its hind legs. Let me take it forward a little to see. Yes, that at this time, let me see if I can pause this. Yes, we can see from this video that the rat was actually standing on its hind legs, and this action is called hinding in behavioral studies. It means it stems from an escape instinct when placed within an enclosed area. So it is expected that a rat that has high cognitive ability or high level of cognition, or display lesser anxiety levels, would make escape attempts, because it's just a natural instinct that animals should have. if it has not been impaired. So we can see the rat standing on its hind legs in an attempt to make an escape from the box. And this is particular, this is very significant, it's a very significant index in behavioral studies. And we use this to measure cognition. We use it to measure how the rat, is not exhibiting any form of anxiety or fear. In other rats that had EM exposure, you could see that they exhibited very low exploratory, activity. They were just staying at one spot, not moving, or the defecation, count was so much by the time of observation was over. So this is a standard method described by Bowen et al. 2014, a standard behavioral method for measuring line crossing, for measuring anxiety in rats. And also the rotating rod, I want to escape from here.
Okay.
So I can.
So they had more defecation and they had less attempts of hinding where they were trying to escape.
Exactly.
So it's like higher anxiety, but less exploratory behavior, less inquisitive.
Exactly. They had higher anxiety. We call it anxiogenic behavior. They had they exhibited anxiety by cringing and hiding away. First, another thing that I didn't mention was that we also look at the number of time the animals spent at the corners of the boxes, in an attempt to hide away out of fear. The rat would go into corner of the boxes and just stay there for some time without making any move, just hanging somewhere around the end, the corners of the boxes, and they stayed there for quite a while without making any move, without making any escape attempt, and this is indicative of anxiety in such rats.
What is happening right now in Nigeria? I know that there was a discussion at the Senate. What is the state, the situation now, and what is the next steps for the country?
Okay, thank you for asking. Based on my interaction with the head of research, Nigerian Communications Commission, NCC, which is the regulatory body for deployment of such technologies in Nigeria. I was contacted through LinkedIn by Mr. Kenneth Nwakor, and he placed some requests for some of my research papers. He requested for them so they could make informed decision as raised at the Senate meeting. So I also included in separate notes some of our findings. I included them in a not very scientific form, so a layman could understand what the consequences of our findings, what they actually mean. So later on, I found out on the news and via different social media platforms that the 5G deployment has been temporarily put on hold, and this is because based on the executive vice chairman's statement, press release on the fact that there was a trial 5G deployment last year, around November 2019, which lasted for about 3 months. And during this trial, I think they also made use of some security agencies and agents as well. And based on their findings, which have not been published or which were not stated, you know, they debunked the rumors that 5G was linked to COVID-19, and they also stated that this 5G has not been deployed. This 5G technology is yet to be deployed in Nigeria, that it has been temporarily put on hold. So we are hoping that they would bring up or suggest more researches to be done in this area. We hope the NCC would publish expression of interest, and calling for different researchers to look into this properly in a scientific way, and not just from hearsays or assumptions, to clear out and also establish the safety of this 5G before it is being deployed for use in Nigeria.
Thank you. I know that what they're doing in the United States, and I think what's happening around the world, is there's a lot of talk about 5G, and there are 5G test networks that are happening in various areas. But what's also happening is the densification of 4G and these cell antennas that are being placed near homes that will in the future be able to house 5G antennas, but are really having the 4G and the frequencies currently and modulations currently in use. So there's research that's shown that's increasing the radio frequency levels in the vicinity of the antennas, of course, but that's also been documented in research. But it's not 5G. 5G can be popped on later. In some areas, they're doing 5G, but of course, we don't have 5G phones that are in use in any wide way, certainly not in the United States. There's 5G ready, or they use the word 5G, but it's not necessarily and always 5G. It's really 4G LTE. So, but it's still an increase in exposure. Is there any kind of public awareness campaign?
Basically, I think for us as a nation here that's still developing, we need more of independent grant opportunities, that helps to, that are university-based, right? Independent university-based grants, so we can put out unbiased and, reports that do not have conflicts, that are not conflicting with interests, both nationally or, internationally and locally. It really helps the research process. It makes it beautiful. It makes it worth the while. It makes it, there's this ingenuity that comes. It makes it real, original, and that's the beauty of research. And I know we'll get there someday. Hopefully, we will get there. We'll get there.
Well, thank you so much for sharing your research with us. And I believe that we will get there, but it's really going to take a level of awareness being raised on how important this is. Because when people first look at it, they say, well, there's no proof of harm. And yet you just provided us with research adding to a large body of research showing harmful effects from everyday devices that we use. Your research you're doing is critical. It's urgent.
Thank you very much. It's my pleasure, and I'm greatly honored to have been part of this. And I'm happy that we are lending our voice. And thank you for, giving us this channel, to lend our voice and make us heard. in a better way, such as to, rouse people's belief and also make them know that they are not mere hearsays, but they are proven. Thank you. Like you said, in the United States, they just need to, there's a lot of, they stand the risk of losing so much more if they don't take note of what the production, how to bring about safer technologies. I hope the regulatory bodies in Nigeria would also make their regulations strict enough to encourage the influx and production of safer technology that has, the people at the center, at the core of its, production, not just for advancing seamless communication or for meeting up with data demand, but have, people's needs, people's health concerns at heart. So I'm happy. I've learned a lot, and I'm happy to have interacted via this platform. I'm not taking it for granted at all. Thank you so much. Thank you.